Okay, section 1.5, it's all about proteins. I'm going to start out by talking about the monomers of proteins, which are amino acids. Proteins are polymers whose monomers are amino acids. The general formula or chemical structure of an amino acid is being shown right here. And at pH 7, the carboxylic acid group will have a negative charge and the amino group will have a positive charge. So to just show this here, so this is what an amino acid looks like, and you have the R group or the side chain coming off of that carbon right there. Now, every amino acid has the exact same backbone except for the side chain being different. So there are 20 amino acids. The amino acids have different side chains. An amino acid molecule by itself is hydrophilic due to the plus charge on the amino acid group and a minus charge on the carboxylic acid group at pH 7. So just looking at this molecule and what we have learned previously about hydrophilic and hydrophobic, well, an amino acid has a minus charge here and a plus charge right here. So the molecule by itself is hydrophilic. It will go into solution with water very well. That is the whole molecule. Let's just keep that in mind. Amino acids are covalently linked to each other through peptide bonds. Peptide bond is an amide bond and is a product of a condensation reaction between the carboxylic acid group of one amino acid with the amino group of another amino acid. So here we have two amino acids, one here, one here. You have a water. Essentially the water is a product of this reaction, and you have a formation of a peptide bond between the two amino acids. We can draw this up in a more familiar way, where we have two amino acids. We know they're two different amino acids because one has the R group and the one is R prime, and that means that these two side chains are different. And this gives us the following, plus an H2O molecule. Of course, this reaction would have an enzyme involved in it. Now the formation of a peptide bond removes the minus and plus charge from the carboxylic acid and amino group respectively. So what all that means is that if we go back and look, this minus charge and this plus charge are no longer there anymore. They've been removed when the peptide bond was formed. In a large polypeptide chain, only the ends will have a charge. These ends are termed the N terminus, the plus charge, and the C terminus, the minus charge. So let's go back and take a look. So you can see that one end has a plus charge, another end has a negative charge. This end would be the N terminus, and this end would be the C terminus. Okay. Amino acids are classified based on their side chains. So the classification of amino acids, let's draw an amino acid right here, is not based on this part of the molecule, but only based on the side chain, the R group. So this classification system is based on that R group or side chain. And we have the polar amino acids, hydrophilic. So these are all hydrophilic. We have polar charged, which means the R group will have either a full positive or full negative. And these include the acidic amino acids and the basic amino acids. And here are the basic. The acidic will have a negative charge on their side chains. So here we go. We have a negative charge here. We have a negative charge here. That's permanent. It will be there at pH 7. And the basic will have a full positive charge. That is being shown here, 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 right? So they all have a full permanent plus charge on them at pH 7. There are polar uncharged groups, meaning that the side chains are polar, they're hydrophilic, but there's not a permanent full plus charge or full negative charge, just partials. So polar uncharged, you can see there's an NH2 amino group here and an NH2 group here. The nitrogen will have a partial negative charge on it, and the hydrogens will have a partial positive charge on them, but there's no full charge. And we have serine, threonine, and tyrosine, and they have OH groups, and we know OH groups are hydrophilic. Oxygen is going to have a partial negative, hydrogen will have a partial positive. These three amino acids are very important to a topic we'll be discussing in section 3. Frequently, when a protein is phosphorylated, it is phosphorylated either at a serine, threonine, or a tyrosine, where these OH groups are replaced by a phosphate group through a condensation reaction. Then you have the nonpolar amino acids. And all these are all the amino acids with a side chain that is nonpolar, thus hydrophobic. So you can see here, you have just a methyl group, another hydrocarbon chains with a sulfide. This one, tryptophan, is kind of weird because it does have an NH group, but this whole thing right here is an aromatic ring. And if you've taken organic chemistry, you know aromatics are weird. So this amino acid, its side chain, is actually nonpolar or hydrophobic. And all the other ones are basically the same way. These are all nonpolar side chains. Thus, these amino acids get classified as nonpolar amino acids. Now, two cysteines can form a disulfide bond, and that's being shown here. We're not going to be discussing disulfide bonds in this class, but it's something to be aware of. So the polypeptide, and let's go ahead and define what this is, a polypeptide. Because sometimes the word polypeptide and protein are used interchangeably, and they can be used interchangeably, but sometimes a protein is not just one polypeptide. So what is a polypeptide? When you have multiple amino acids that are linked together through peptide bonds, and that is being shown here, for example. Here we have 4 amino acids. One amino acid right here, one amino acid right here, third here, fourth here, right? And they're linked together through peptide bonds. Now, this is a single chain of amino acids, and that would be a polypeptide. If that single chain of amino acids eventually folds into its three-dimensional final structure, its native structure, we can say that this functional protein now has a single polypeptide. It's a single polypeptide chain. Just keep that in mind. When we say polypeptide, we mean just a chain of amino acids. And that chain of amino acids can fold into its three-dimensional structure and be the functional protein, or the polypeptide. They're interchangeable in that case. Now, the polypeptide, that single chain of amino acids, minus the side chains. So, let's go ahead and remove these arrows real quick. So this thing right here is the polypeptide backbone. And the side chains are coming off of the polypeptide backbone, of course, the side chains being covalently attached to each individual amino acid that is part of the polypeptide backbone. Now, this picture also gives you a really good idea of where the 4 amino acids start and end. So, for example, this is one amino acid's backbone, and this becomes the full amino acid right here. Here is the second amino acid right here. Here is the third. And here's the fourth. So to minimize all the writing and everything that needs to be done and show every carbon and hydrogen and oxygen and nitrogen, typically when we try to write out a polypeptide, we just write out the code. And if you go back, and we'll go back, and look at our amino acids, you can see that there's an abbreviation for each amino acid. For example, phenylalanine, the 3-letter abbreviation is PHE, and the single letter abbreviation is F for phenylalanine. Glycine is GLY and G. So when you're writing out a polypeptide, instead of putting all the carbons, oxygens, and nitrogens and hydrogens, you can simply write out the code like it's being done right here. Of course, they're showing you the full name and the 3-letter abbreviation underneath, but you can simply write out the 3-letter abbreviation. MET for methionine, ASP for aspartic acid, leucine, LEU for leucine, and TYR for tyrosine. The way these are written out is that you always start out with the N-terminal side on the left-hand side and end with the C-terminal side on the right-hand side. So it's always N-terminal to C-terminal, and that's what's being shown here because it's directly linked to the picture above, right? So it's always N-terminus to C-terminus. So classification of protein structural information. And just to make sure we understand what this means. These classifications of primary structure, secondary structure, tertiary structure, and quaternary structure, all this is giving you is information on the structure of the protein. And the structure of the protein is complex, so the information is being broken down into these 4 parts. So primary structure would be the sequence of amino acids from the N-terminus to the C-terminus, basically giving you that first bit of structural information of, okay, what are the amino acids and in what sequence are they chained together from starting from the N-terminus to the C-terminus. So that's the first bit of structural information. The second bit of structural information is what local structures are within that protein. And the most frequent and the ones that you probably know of already are the alpha helices and the beta sheets. So alpha helices, and down here we have the beta sheets. So what are alpha helices? And here's a picture of an alpha helix, and you can see the polypeptide backbone kind of right here. And this is not showing you the side chains in its full glory. It just has them written out as R groups, and you can see that the R groups are coming out of the alpha helix. So they're sort of on the outside. You can see one's here, one's here, one's here, one's here. Right, so these are the side chains. The alpha helices are being formed by hydrogen bonding between the carbonyl oxygen and the amide group of the polypeptide's backbone. So when we're writing out our polypeptide chain, so here we go, we'll do a tripeptide, 3 amino acids. And we have our R groups. So what we mean by that, you have hydrogen bonding between this carbonyl oxygen and this nitrogen right here. So you can see here that this hydrogen bond being formed right here, right here, right here, and etc. from these dashed red lines, it's between the hydrogen of the nitrogen right here. You can see this is the hydrogen right here. I'm going to kind of highlight it in red right there. And the oxygen, and the oxygens are the red balls, and the oxygen of the amino acid above. And that is stabilizing the structure of the alpha helix. So in an alpha helix, a hydrogen bond is formed between every 4th peptide bond between the carbonyl oxygen and the amide hydrogen. So every 4th amino acid, the carbonyl group, is hydrogen bonding with the amide group. To rephrase that, amino acid 1's carbonyl group is hydrogen bonding with the amide group of the 4th amino acid downstream. Now beta sheets are again very similar in how they're formed. You still have hydrogen bonding between the amide hydrogen and the oxygen of another amino acid, the carbonyl oxygen of another amino acid. So you have these polypeptide chains being side by each other, and this is being shown in this picture right here very clearly. And what this sort of arrow thing here means is that if this is the N terminus and this is the C, then this one is the N here for this one, and this is the C for this one, N for here, and C for here. Okay? So they're running anti-parallel to each other in this picture, meaning that one polypeptide chain is running in one direction while the other one is running in the opposite direction, anti-parallel, although they're right next to each other. Now you can see that, again, the same type of coloring is being used where the oxygens are red in this picture right here. The oxygens are red and the nitrogens are blue, and these are the hydrogens right here of the nitrogen. So you have hydrogen bonding happening between the carbonyl oxygen and the amide hydrogen between the chains that are passing by each other. And that keeps these chains close to each other, thus forming the beta sheet. So each single one of these strands, each single one of these polypeptides that are part of the beta sheet are called beta strands. So for example, this right here is a single beta strand. And the next one over is another beta strand. And the one next to it is another beta strand. And these beta strands form the beta sheet. And a beta sheet can be many beta strands or very few. But the overall structure looks like this. The other thing to also notice is that the R groups, the side chains, are sticking out from the beta strand. They're either sticking out of the page or going through the page and they're coming out the other side. So if I was going to look at a beta sheet from the side, so here's my beta sheet looking at from the side, the R group would be sticking out, alternating between coming out on one side or coming out the other side, like that. Okay? So beta sheets are composed of beta strands, where each strand's polypeptide backbone is hydrogen bonding with the two strands flanking it. So for example, in this picture, the middle strand right here, so this would be the middle strand, would be this one right here, it's hydrogen bonding with both the strand to its right and the strand to its left. Okay? That's what that statement means. The carbonyl oxygen from one strand is hydrogen bonding with the amide hydrogen from the adjacent strand of the polypeptide backbone, of course. The beta strand might be connected to another by a short random loop or other secondary structures flanked by a random loops. So what that means is, for example, this picture is showing you a beta sheet, but it's not showing you the rest of the protein. So the rest of the protein could look something like this, where I have one beta strand, and an arrow like this is just a shorthand way of showing a beta strand, being attached by a random loop. This is the rest of the polypeptide. And you get the second beta strand. Right. You can have another sort of the loop, and I'm just going to put a little tail for this one right here. And this could be attached to, let's say, an alpha helix. Typically, an alpha helix can be shown as a cylinder. So I'm just going to draw a cylinder. Okay, so I have an alpha helix, and then I have kind of a little bit of a loop coming back, and I get another beta strand. Right here, and then off it goes, right? So For example, if I was going to explain the secondary structure, let's say this is my protein right here. I can explain the secondary structure by saying that, well, this protein has one beta sheet composed of 3 beta strands and a single alpha helix, plus a couple of random loops. Okay, so that's giving me information about this overall structure of the protein. That, okay, it has a beta sheet that has 3 beta strands and it has an alpha helix. Now I can get a little bit deeper and say that, okay, it has a beta sheet where 2 beta strands are attached to each other by a random loop right here, and another beta strand is part of the beta sheet, but there's an alpha helix between them right here. So that gives me a little bit more information about the structure of the protein. So beta strands can be running parallel or anti-parallel to each other. And this is showing you the anti-parallel version, where you're starting out with, if this is a single polypeptide chain, which this is what it's showing you, it's a single polypeptide chain. If this is the N terminus right here, and this is the C terminus, then coming back, this would be the N terminus, this would be the C terminus of this beta strand, and this would be the N terminus, and this would be the C terminus of this beta strand. Okay? Now, in the case of parallel, you have these long loops connecting the beta strands to each other as part of the beta sheet, and if this is the N terminus and this is the C terminus, and I come back, this is still the N terminus and this is the C terminus and this is the N terminus and this is the C terminus. But of course, there's a long chain of amino acids connecting these 2 beta strands to each other. And you can have a case where it's not just a single loop, you have another secondary structure in between, and then you get the next strand. So if I was going to draw this out, it would look something like this. So you could have it be something where you're coming all the way back to start this, right? But instead of coming all the way back like that to start the second beta strand, so they can run parallel to each other, what you can instead have is it coming back, you have an alpha helix, and a loop connecting to the next beta strand, part of the beta sheet. And then you can of course have 2 running parallel to each other, followed by 2 running anti-parallel to each other. That is completely okay. Again, that's secondary structure information. In random loops, a string of amino acids with a variable non-static structure, always in the aqueous solution. You will have a better understanding of why we're saying that random loops are always in aqueous solution later. But typically these random loops don't have a static structure, or they're not very structured compared to alpha helices and beta sheets, where that structure is kept as part of the native structure of the protein. Tertiary structure. The full 3-dimensional organization of a polypeptide chain is the tertiary structure. In general, for any polypeptide chain, its stable conformation in the intra or extracellular space is generally one that minimizes its free energy. So just the way we had reactions and we had a delta G, you can have a protein in a completely unfolded random state. So we're just going to say unfolded. So UF for unfolded, which would be a high energy state, and it will move down to the lowest energy state possible, and that will be its folded state, so F, where it will achieve its tertiary structure. Now, this difference between the folded and the unfolded state, well, there's a delta G for that, right? That is its delta G, the delta G of folding. So the folded or the final shape of a single polypeptide chain, this is very important because only a single polypeptide chain can have a tertiary structure. So that tertiary structure for that single polypeptide chain, that folded, final folded state, is going to be a low energy state compared to the unfolded state. Quaternary structure. So many proteins are not just composed of a single polypeptide chain. they're composed of multiple polypeptide chains coming together to form a bigger structure. And that structure is the final protein, meaning that as a single polypeptide chain in its tertiary structure, that single polypeptide chain in its tertiary structure does not have biological activity once it is part of a complex with other polypeptides that have formed their tertiary structures. It becomes the functional protein. An example of this is the hemoglobin protein. It's an example used frequently where you have two HB beta subunits and two HB alpha subunits. And these are attached to each other through weak interactions, and this forms the hemoglobin protein. By themselves, the HB beta or the HB alpha do not have any biological activity, but they are single polypeptide chains, and each HB beta and each HB alpha has a primary, secondary, and tertiary structure. But the protein hemoglobin has a quaternary structure. HB beta, HB alpha don't have a quaternary structure, but the protein hemoglobin does. And what is the quaternary structure? It is two HB alphas and two HB betas. And that is the structural information about hemoglobin. Now, if the question was, what is the primary structure of hemoglobin? Well, hemoglobin itself does not have a primary structure. HB beta has a primary structure. HB alpha has a primary structure. Hemoglobin does not have a primary structure. What is a secondary structure of hemoglobin? Hemoglobin itself doesn't have a secondary structure. HB beta and HB alpha have secondary structures. What's a tertiary structure of hemoglobin? Hemoglobin does not have a tertiary structure. HB beta and HB alpha have tertiary structures. So the primary, secondary, and tertiary structures are structural informations only for single polypeptide chains, where the quaternary structure is information of whether the functional protein has partners, it's many polypeptide chains coming together, or is it just a single polypeptide chain? Because if a protein is just a single polypeptide chain, its quaternary structure is just that single polypeptide chain. One last but important note. The final biologically active shape of protein is generally called the native structure, but not all proteins have a single native structure. Probably up to this point, you have only encountered or only have been told that proteins have a single native structure, but that is not true of all proteins. Some proteins have multiple native structures and will switch between them in a random, probabilistic manner. The likelihood of being in one conformation to the other is dependent on the stability of each state. So for example, let's draw out that folded and unfolded state. So we could have unfolded, unfolded. We'll just put unfolded priming. This unfolded state is a little bit different than the other unfolded state. And we could have the folded native state 1, folded state 2, and folded state 3, and if they're basically at the same energy level, what will happen is the protein can be in any three. It'll just kind of switch between them. Now, just like a chemical reaction has an activation energy barrier, so does the switching out between the three states. So if I was going to draw out a path from unfolded to folded, you can imagine it will look something like this. You go to from unfolded to folded state 1, there might be a little bit of an energy path where the protein has to unfold a little bit to get the folded state 2, and it needs to unfold a little bit to get the folded state 3. And then, of course, here. So, in this case, this example that we have here, the way it's drawn, the protein has to go from folded state 1 through 2 to get to 3. From folded state 3 to folded state 1, it has to go through folded state 2. Now, these are relatively at the same energy level on the bottom right here. So it is just as likely to find the protein in any of these three states. One is not favored over the other. Now, if I had the case where folded state 1 was a little bit higher, and so was folded state 3, and I have this pathway here, let's draw it out. Boom, down to here, a little bit of a bump, down to here, a little bit of a bump, up to here, and then down. Then folded state 2 is going to be preferred over folded state 1 and 3, but since there's such a narrow range, right, between them, that difference in energy is so little that because of the temperature of the solution and just random thermal motion, that the switching will occur, but the protein will be in folded state 2 more frequently than it will be in folded state 1 or 3. Okay, so just something to keep in mind. The kinetics of switching between these different conformations is dependent on the energy barrier akin to energy of activation between each state. And we kind of drew that here where we have sort of these energy barriers right here, for example, and right here, for example. And if they're rather large, how fast the protein switches between one state and the other would be slower. Whereas if those barriers are a little bit less, it'll be faster. Some proteins will change their native structure based on environmental changes such as pH, temperature, pressure, light, and voltage. So that means that the proteins are not stuck in their native structure. They might be in one native structure at a certain pH and switch to a different native structure in a different pH. Now, why would a protein do that? It could be that, for example, at an acidic pH, a little bit less than 7, it's in one state where it's inactive, whereas when the pH goes to 7, neutral or higher, it goes to a different state where it's active. This does not mean the protein unfolds, it just means that it has two different states. And the biological activity of the protein will change when switching from one state to another. The same thing with temperature, pressure, light, and voltage. Some proteins will change their conformation when covalently modified. A common example of this is phosphorylation of serine, threonine, tyrosine side chains. For example, we have an enzyme that is inactive when in its non-phosphorylated state, but when it's phosphorylated, of course ATP to ADP, it is active. So when it's phosphorylated, there's a small change in its native structure. So now it has a different native structure, and that native structure, that new one, will be the active native structure. Whereas in the inactive state, it was in a different native structure where it could not catalyze whatever reaction it's supposed to catalyze. Now again, it does not mean that this state is unfolded. This is not the case. Unfolded means something completely different. Okay, so don't think that that inactive state is an unfolded state. Okay? And we will see examples of all three types throughout this course, and that is why we're talking about these things. Okay? Alright guys, we'll stop here and continue later. Bye-bye.